Hi, everyone, and welcome back. Now we're going to enter the regulatory portion of our day today. And first, we're going to hear from two experts at the FDA, Gautam Mehta, who is a neurosurgeon and also in Division of Neuro-oncology at the FDA as a reviewer, and Greg Clement, who's in OSL, the Office of Science and Engineering Laboratories, a research physicist with an expertise in ultrasound. So they're both going to provide a presentation, and then several others will join us for a panel discussion.
Hi, good morning. Thanks so much, Jessica and the Focused Ultrasound Foundation for inviting us to speak. My name is Gautam Mehta. I'm a neurosurgeon and clinical reviewer in the Center for Drug Evaluation and Research at the FDA. I'll be presenting with Greg Clement, who's a reviewer in the Office of Science and Engineering Laboratories, or OSL, in the Center for Devices and Radiologic Health. Today we'll be talking about the FDA perspective on early stage combinatorial trials using focused ultrasound. We can go to the next slide, please. We have no disclosures to report. Next slide. So today we'll review how oncology at FDA is organized and its role in drug development, as well as organization of the Office of Science and Engineering Laboratories. We'll also go over project jurisdiction and how that may apply to FUS studies using drugs. Since many of you may be more familiar with the IDE process for devices in CDRH, we'll review here the investigational new drug or IND application process for applications that come to the Center for Drug Evaluation and Research, or CDER. We'll also talk about, we'll talk about this IND review process, provide some general advice, and then finally we'll discuss some device-specific considerations for these applications. Next slide, please. So the FDA is comprised of 3 centers: the Center for Biologics Evaluation and Research, or CBER, the Center for Drug Evaluation and Research, or CDER, and the Center for Devices and Radiologic Health, or CDRH. In oncology, these 3 are brought together through the Oncology Center of Excellence that promotes a unified vision across centers for cross-disciplinary collaboration. This Center of Excellence, or the OCE, is comprised of over 100 oncologists, in addition to other specialists and experts. This is the first and only inter-center Center of Excellence at the FDA, and was started in 2017 as part of the Biden Moonshot Initiative. This cross-disciplinary collaboration helps us to more seamlessly review protocols or applications that are multidisciplinary, such as trials using FUS for blood-brain barrier disruption. Next slide, please. At the macro level, regulatory review in oncology uses a multidisciplinary approach to think about how we advance patient-centered regulatory decision-making. So this includes regulatory research and science here at the FDA, internal and external educational programs and outreach such as this, stakeholder engagement with groups such as SNO and NBTS, and of course work on regulatory policy and law. Can we go to the next slide? And here I'll hand it over to Greg Clement to talk about OSL.
Great, thanks so much. So as Gautam looks out the window from the beautiful building of the Center for Drugs, he looks across at the little bit more modest world of the Center for Devices as we stare at each other. And I'm coming from the research end of the Center for Devices, and that is OSL. And so as we kind of see a summary here, one of the main take-homes is that in one sense, we operate very much like an academic lab would. We have students, we have postdocs, and we do regulatory research. Another big piece of that, as we see down by the 2,500 so-called consults a year, that we are doing scientific reviews into the products that come into the Center for Devices and Radiologic Health. Now, OSL restructured in 2020 into a group of programs, and we see the list of programs set up here. And these were designed in these 27 labs to center on where we saw the current need in regulatory research and the need in the near future for regulatory research. And so one of those programs that came out of that was the program for therapeutic ultrasound. And so that's sort of a nice testament of where the field has been going, and the fact that the FDA saw it enough, importance enough to give it an individual program. And now from here, if we go to the next slide, it's just a sort of an illustration of the research and how we work with the different centers. So coming from devices, I'm personally doing research primarily in transcranial ultrasound. Matt Myers, who is here today, is also doing a lot in terms of cranial ultrasound. And so much of the work that we're doing is in collaboration with CDER and CBER, the Center for Drugs and Biologics. And so focused ultrasound, in particular transcranial focused ultrasound, is certainly an area where we are not in a silo. That we have an area for physicists and engineers that necessarily have to be working with clinicians as well as biologists and chemists in looking at this problem. So we go to the next slide. And part of what we do is from the very basic end of ultrasound and acoustics, and that is looking, when we have these devices, on methods for how we can characterize them. And folks coming from the industry world, these are very familiar looking methods. And so we look into ways that we can calibrate and very accurately measure power, acoustic fields, and just so that we can know the output of these devices. Now, going on to the next slide. I said that you know, this is an area that in research we have to reach out for collaborations. Certainly when it comes to the products themselves, we have in much of transcranial ultrasound devices that either don't fall into a category or more commonly fall into more than one category. And we run into the world of combination products. And many of you are familiar with the IND process, many are familiar with the ID process. And what I would say is certainly at these early stages, the main consideration from the FDA is safety. So in many respects, the review process that you would go through, whether you go through an IND or an IDE, would feel very similar. If we are working through one center, that is, for example, through CDER, the device side of the product would be looked at still through CDRH. And so regardless of what the lead center is, the entire FDA or the entire relevant portion of the FDA is involved. If you look at the next slide, the big question then comes down to who is the center that would actually do a review? And that actually can be a somewhat complicated question. As I mentioned, in terms of the review, particularly at the trial stage, the actual review process might not be that different. However, once you are given a jurisdiction into a center, so it's CDRH or CBER or CDER, this is going to be the center that will carry out the regulation process throughout, all the way to the point of marketing. And so there are questions that come up in terms of what center something could fall into. In terms of transcranial ultrasound, and particularly transcranial ultrasound that also involves microbubbles, it also involves a drug. It's not necessarily a directly straightforward question to answer. And in fact, in terms of our Center for Combination Products, it sort of puts on a clinic in terms of determining what area things will fall into. And so this is when you have a new product, an area where if you are not sure, we would encourage you to actually reach out to the Office of Combination Products at the FDA and can have a discussion and start a dialogue on what can be done in terms of having your device or device and drug fall into a given center, which will be based around, in the end, the primary mode of action. And so now I think I'm going to pass this back over to Gautam.
All right, thanks, Greg. So now we'll get into a little bit of the nitty-gritty of our review process from the viewpoint of an application that comes to CDER as an IND. And so our regulatory review at the individual application level is also grounded in that same multidisciplinary approach that I spoke about earlier. So this includes project managers who would be your point of contact during and after an application submission, experts in preclinical or animal work and toxicology, experts in product quality, so how the drug is made, if that's relevant, clinical pharmacologists, clinicians that may have varied in specific specialty expertise, such as neuro-oncology, radiation oncology, and surgery, biostatisticians with expertise in clinical trial design, and of course, engineers and clinical device experts and clinicians in CDRH. Can we have the next slide, please? Regulatory review at the FDA spans the breadth of product development. And in CDER, that starts with the IND application for products that come to us with a new drug. This application is typically required before the start of any drug trial in the US. And its primary purpose is to protect clinical trial participants and to evaluate the quality of science in later phase studies. Because this is what's most relevant to early stage research, such as FUS trials, we're going to focus on this topic today. Can we have the next slide, please? When you submit an IND, it undergoes a 30-day review, which is focused again on patient safety. So this determines if the IND is classified as either safe to proceed or placed on hold. And we review the IND to make sure that it doesn't pose any unreasonable risk of illness or injury to patients, and that the study is adequately designed to meet its objectives. For early stage studies, these objectives are often identifying a maximally tolerated dose or a recommended phase 2 starting dose, or recommended phase 2 dose, or identifying the safety and tolerability of a drug. Can we have the next slide, please? So we pay special attention to the patient population that's proposed and consider what are the available or alternative therapies for these patients. Is there a standard of care? So for glioblastoma, that may be different between newly diagnosed and recurrent populations. We look at the seriousness of the disease, which glioblastoma certainly fits, and any known toxicities that may be relevant to this population. Finally, we identify any special populations being treated, such as children or pregnant women. Next slide, please. We also pay very close attention to dosing and how the dose is escalated in early phase trials. We review if the proposed dose is safe. For example, if there's prior human experience, we consider that in the dose selection. If there's not, we strongly consider the toxicology or preclinical data or the animal work. If you're submitting a dose escalation study, we also think about how rapidly that dose is being increased between cohorts. So for biological drugs, this is typically log or half log increments. For small molecules, this may be fixed percentiles or follow another specified pattern like a modified Fibonacci sequence between cohorts. Finally, intra-patient dose escalation can be done and used in these early phase trials. But this needs to be considered very carefully and monitored for any specific toxicities between escalation. Can we have the next slide? In typical phase 1 studies, dose escalation and study endpoints are determined by dose-limiting toxicities, or DLTs. These are typically the primary endpoint of the study and what you're looking for to determine that maximally tolerated dose or the recommended phase 2 dose. There are a number of dose escalation schemes, including a standard 3+3 design, but more and more we're seeing Bayesian designs which adjust to a specific DLT rate and target that. These DLT criteria should be very clearly defined in the protocol, and any unusual criteria, specifically the exclusion of specific adverse events, should be justified thoroughly in the protocol. Finally, when you're considering DLT, the context is important. For FUS studies in glioblastoma, these are patients with very serious cancers. Also, in what setting are the DLTs being monitored? And then finally, for certain drugs, the observation period for DLTs may have to be extended. in the studies that we review, this can be important for continuous dosing trials, which may be less relevant for the focused ultrasound combinatorial studies. But in particular, it's very important for drugs which have a longer half-life, or immunotherapies which can have lasting or late adverse events. Next slide, please. This safety monitoring is really critical to the IND review process and what we look at during our review. In the protocol, you should provide a calendar of events with appropriate testing for specific risks. For example, EKGs should be scheduled if prolonged QT intervals are a concern in the study based on the preclinical or earlier clinical work. Pulmonary function tests, as another example, should be specified if pneumonitis is a concern. First in human studies have a risk of unknown toxicities and often require more frequent monitoring in labs. And in general, for these kind of drugs without significant clinical experience, we really consider that preclinical or animal data. As before, consider the type of drug and its half-life, as this may affect the safety monitoring plan. Next slide, please. Finally, we'll also review the informed consent document that you submit. And for these, it's just important to be honest and transparent. That means not overselling benefits or minimizing the risks of therapy. For glioblastoma studies, often a relevant risk that we see frequently overlooked is the potential that therapy may delay surgical resection or radiation therapy, specifically if these are part of the expected treatment plan for these patients. Finally, any alternative therapies or standard of care should be detailed in the document. Next slide, please. If you're going to submit an IND application, you may want to consider requesting what's called a pre-IND meeting first. And that's especially if a study is complex or has any unusual features. And studies that use combinatorial therapies like FUS studies could certainly qualify for that. These meetings are scheduled within 60 days of your request and typically include the team that will be eventually reviewing your formal IND application. After your request for this meeting is put in, we'll either reply with a written response or schedule a telephone conference. To get the most out of these meetings, we recommend you submit questions related to very specific issues or complexities in the trial that you want us to respond to and want advice on. Also, for the specific methods or issues that you want addressed, it's important that you provide thorough background information or a complete protocol. Next slide, please. So just some general do's and don'ts for the IND application. Do provide a justification for the chosen dose. Provide an adequate monitoring plan. Do expect comments from us that will require a quick turnaround in that 30-day period. And consider asking for a pre-IND meeting if the study is complex. After you submit an IND, don't go off the grid in that 30-day period. We may need to contact you if there are any issues with the study. Otherwise, you should plan to designate an authorized contact who can speak for you and answer questions regarding the study. And then finally, it may be tempting to copy and paste from protocols of other studies, but avoid including any irrelevant or incorrect information in the protocol. And the next slide. I'll hand it over to Greg and now to talk about device considerations and wrap things up.
Great, thanks. Yeah, targeting in on the device part of the review, there is, I'll say, extra challenge to ultrasound device companies in that with transcranial ultrasound, there are no existing standards to follow by. Now, there are HIFU standards, and they're very nice, clear paths that are FDA recognized that would let you characterize your HIFU device. Now, in terms of looking at the characterization of fields and more importantly, what parameters you should be using and whether they're safe or not, it is up to the submitter to show that the device is in fact safe. Now, from the standpoint of ultrasound safety, we're typically looking at the basics of mechanical safety, thermal safety, and so they say the combination of thermal and time, or the notion of dose or the amount of energy that's being deposited. And so to do this, we are looking for data that will show that the target area is the area where we have either a lethal dose or the amount of energy that you want, and we're nowhere away from the target has any possibility, even under worst case scenarios, of causing damage. Now, as I say, there's no standards for actually picking these parameters. At the same time, there are some standards that can serve as very useful guides, and I'll go back to that in just a moment. But if we could have the next slide. In general, I would say for device considerations, we put a list here together of things that we would nearly always look for when an ultrasound device, a particular therapeutic ultrasound device, comes into the FDA. In short, what we'd like to know is we'd like to know calibrated what is the actual power that is being put out and in what sequence. And so in addition to that, we would like to see the characterization of your transducer. What does the field look like? But I'll say in the beginning, and just anecdotally, one of the things that I see missing quite often is just literally the absolute power, and the geometry of your transducer immediately can tell us a lot about what your device is doing. So the general operation parameters in the field are something that we look at. With transcranial ultrasound, there is the challenge of looking at the worst case parameters after going through the skull. And so what I would say is that in reviews, it's particularly a good idea to look at very extreme conditions. For example, if you're past the focus and you're going into bone, what would happen if all of the energy, for example, that goes past the focus were absorbed by the bone? If that would not lead to a safety issue, then I think the physics of the problem has already shown safety in that incident. If it's something that's marginal, then it's something that you might simulate or want to take actual data with thermocouples to continue on and give us data that indicates safety. Now, on the next slide, I mentioned a second ago that there are no standards, but there are related standards that can be useful. Some more useful than others, depending on your application. Blood-brain barrier applications where pressures tend to be low often might find some guidance within our physiotherapy and diagnostic guidances. And again, these make good references. However, ultimately, what we'd like to see is a demonstration or data that demonstrates that your device can operate safely. Now, one mechanism that the FDA has been looking into to try to make that process a little bit easier, as you can see on the next slide. If we go to the next slide, please. Here, is what's known as the MDDT, or Medical Device Development Tool Program. And so what we're soliciting actually is from industry, from academia, from organizations, any type of technology or tools that might make the review process for FDA a little more streamlined. In one respect, this is something like a standard, but with the idea that this in fact could be a technology such as, a concrete example would be a phantom, or some methodology that could be looked at that, if done in a very given and stated way, would help streamline the FDA review process once that tool has been accepted. And if we go to the next slide, I just want to take a summary of what we talked about here today. And so we took a look at the fact that clearly the regulatory process is multidisciplinary, it's collaborative, it spans literally at times the entire FDA. Products, depending on how the cards fall, can end up as an IDE or an IND. Regardless, CDER and CDRH for transcranial ultrasound devices, particularly in the brain involving a drug, would be something that both CDER and CDRH would be involved in. So for IND applications, you can request a pre-IND meeting. We'll add in, certainly with CDRH, you can also request a pre-meeting with CDRH. Reviews focus on safety, that's regardless of center, and any device that's being used, again, regardless of whether it's going through an IND or an IDE, is something that's considered investigational and will be reviewed for safety within the context of use. So if we could go to the final slide. I just want to give some acknowledgements, both for the group at CEDAR and CDRH. And we have some references here for some guidances that could be helpful. Some contact information with the caveat to say that please, if you do have questions, don't hesitate to contact us. Whether or not we have the answers, I think we tend to be very good at getting you in contact with the people that do know the answers. And so I think we're going to transfer over to a panel here, and I look forward to a very interesting discussion this afternoon.
Okay, thanks. That was great, and I think provided a lot of really helpful information for all of us. Now I'm just going to briefly introduce our panelists. Their bios are all in your program, so I'm not going to give you much information other than to say they're all great reviewers at the FDA and experts in either focused ultrasound or in clinical oncology. So we have Amy Barone, who's a clinical team leader. We have Gautam, who you met, Greg Clement, who you've met, Matt Myers, who's a research physicist also in the OSOL Therapeutic Ultrasound Group, and Bennett Blumenkopf, who's a medical officer at the FDA. And that should be all of us. Okay. So we have actually received quite a few questions in the run-up to the meeting. We put a survey out, and we got a lot of questions there. So we have, I think, a lot to go off of for this discussion, but kind of building on some of what Gautam and Greg, you both were talking about in your presentation. I wonder if, through all that you've seen so far in terms of BBB opening applications with focused ultrasound, these combination drug device clinical trials, is there anything specific, maybe beyond what you had in your presentation, that you can add to the group in terms of things like, baseline pharmacokinetic data that might be needed, this idea of, for a combination drug device clinical trial, do you need to have preclinical data that's using the exact same device and drug combination as you're going to use in your clinical trial? Maybe just speaking to kind of where we are in developing or getting closer to having some sorts of standards for these focused ultrasound BBB opening combination trials. And I'll let anyone handle that or start with that. Okay, Gautam, do you want to start? Or Amy, go for it. Yeah.
Hi, nice to meet everybody. My name is Amy Barone. I'm on the drug side of things with Gautam. I'm one of the team leaders on one of the teams in the Division of Oncology too that reviews the drugs for brain tumor drug development. I'm a pediatric oncologist by training. That was a lot of starting points we could go with, Jessica, so maybe I'll just tackle a couple of things. First, I think I first would say one of the points that Greg brought up at the end was really great, that we're in constant communication with each other between the centers. And so no matter where the trial falls, if it comes to the drug side or the device side, we're going to be consulting the other one to help us through this and make sure that we're providing you with the best advice that we can. And that in the early stages, we're really focused on safety, as was emphasized in the talk. I guess I could comment a little on the non-clinical data. We did reach out to our colleagues on the drug side of things to see what they would require and want to see in one of these kind of drug trials. I think one of the things that they recommended is really looking at evaluating the exposure of the drug administered in the brain before and after plus is administered to the animals. And this might be different depending on how much you know about the drug. If it's a first in human drug or you have absolutely no clinical data, you may want to look at different dose levels, particularly high dose levels before and after in the animals. If you have quite a bit of experience with the drug, you may be able to narrow that focus down, perhaps do a pilot study with a control set of animals, and also making sure to assess the impact on CNS toxicity to the degree that you can. In terms of using the exact same drug and device from the drug side, I think that would be ideal to use the same drug that you're going to use in the clinical trial. But we do recognize that not all drugs are relevant in various animal models per se. So if that drug isn't going to hit the target in the animal, it's reasonable to use a type of treatment that would be more relevant to the animal model that you're looking at, and we would be open to that. I think you would just need to describe what you were doing and why you chose to use that drug. From the device side, I think, I'll let my CDRH colleagues comment on that part of it, but I would think, we would want to see something similar or something demonstrating that the blood-brain barrier is opening and what differences you might be observing. And I'll leave the others to comment.
Hi, this is Ben Blumenkopf. I'm a neurosurgeon. I've looked at a lot of these submissions, particularly with regard to the microbubble blood-brain barrier. And I want to emphasize the usefulness of the pre-submission that was mentioned earlier. Just like in the IND, in the IDE process, we have a pre-submission, which really allows us to open a dialogue and work a lot of things out in advance of a formal submission. And a few years ago, I was on this symposium, and just to reemphasize that if it is a combination between a drug and the device, that you don't submit both, you submit one or the other based on the jurisdiction. And as Amy just said, we collaborate extensively in that regard. With regard to the ultrasound and the microbubbles and the blood-brain barrier, one of the areas that we've been looking at carefully over the years is the volume of that process, such that initially we wanted to see not only that the barrier could be opened safely and then closed over time, period of time, but exactly incrementally allow that volume of opening to increase gradually to make certain that a too large volume that was opened was not in any way a safety concern. So that was one area that we worked carefully based on some preclinical data to support the actual volume of the blood-brain barrier disruption in the brain and allow that to proceed gradually in the human studies. And again, we rely very heavily on our basic science colleagues with regard to the power or the strength of that ultrasound with regard to the bubbles. We've had other issues with changing concentrations or doses of the bubbles that we've had to consider in addition to the actual ultrasound parameters. So again, we encourage the pre-submission process both on the drug side, but certainly on the device side, to allow us the opportunity to get everyone involved deliberately and then to proceed interactively to get the process really to a point of good fruition when the formal submission comes in.
Thank you. That was really helpful. We have Adam Sonnabend here, and he would like to join and ask a question to the panel. So can you please allow him to come up and join?
Hey, how are you? First of all, thanks so much. This is an amazing, very educational talk for me. I guess my question on the big picture is now for trial design and regulatory path, getting preclinical tox data, and moreover in the future for approval of a blood-brain barrier opening device. Do you guys think there's a path to decouple the device and procedure from the drug use? In other words, if you can get a device that can open the blood-brain barrier and you can prove that it does that and then enhances the drug levels in the brain, does that approval need to come in with a specific drug?
Hi, it's Bennett. In general, I would say that each time we're asked to review a device that's then going to have some sort of interaction with a drug, we look at that individually, such that it's not necessarily true with the ultrasound, but there are a number of devices that interact, directly, and we have concerns with compatibility with the devices as far as that drug is concerned. And ultimately, it also relates to how the device and then the drug would be marketed because of this interaction between the device and the drug. And that gets really complicated sometimes, and I have to defer as the clinical to the regulatory people. The other issue that comes up, in my experience, is what the study is actually designed to do or to show when we're talking about devices just in general. But we've seen this with the ultrasounds. And what I mean by that is if you're trying to look at the device as a tool, that's appropriate at times, and a number of devices are approved or cleared as tools, meaning they do something, like an ultrasound, say an ablation or opening the blood-brain barrier. But if then you're going to engage with a drug, and certainly this is also true with the device independently of a drug, but when you're talking about a drug, I think there's a clear implication that that's going to be a therapy. And when we're looking at devices used in a therapeutic context, as opposed to just performing a tool process. So an ablation is a tool process, but say ablation for Parkinson tremor might be a treatment context. Then we look at those trial designs somewhat differently. And again, that's why it may be have to look at it individually. So again, we generally don't like to just clear things or approve things as in that generic tool when the intent is ultimately as a treatment, which, and again, a number of specific drugs with the tool, even if it's a tool, we look at individually generally. I hope that answers your question.
This is Matt. I just wanted to add that I could see that down the road, the ultrasound could be used as viewed as a tool, and that you could have separation between the devices and the drugs. But I think we have to remind ourselves that the microbubbles are involved as well, and we just don't know enough about how the microbubbles, behave in the presence of ultrasound to have a general tool type of view of that interaction right now. So I think we're quite a ways away from being able to separate all of the different components.
So building off of what you were just talking about with microbubbles, obviously microbubbles are required for all of these BBB opening procedures. Are there any guidelines or standards for what you want to see about the microbubble itself, or whether it's, obviously size and dosing and things like that, but is there anything about the ultrasound's interaction with the microbubble that should be in the in the application or should be studied preclinically? Maybe Greg or Matt, do you want to?
Yeah, well, I know that the level of BBB opening and the duration is related to microbubbles. I think we were talking about that yesterday, and I think that the patterns just aren't that well known, at least not by us at this time. So I think the more that we can understand about how different sizes and even constituents of microbubbles open, to what extent and for what duration they open the blood-brain barrier. That's something that we would really like to know. And Greg's doing a lot of work in that right now, and I know that a lot of other people are as well, but that's a biggie for us.
Jessica?
Another question that just, oh, sorry, Gautam, go ahead.
Problem. I just wanted to loop back around to Adam's question. I think, from a drug standpoint, beyond that interaction between the device and the drug that Bennett spoke about, we still would expect an increase in CNS exposure with some of these drugs that's beyond what is currently known or has been currently studied. So there's need for more information at this point.
Great.
And I would just add quickly, sorry, Jessica.
That's okay.
To that, when we're thinking about, I think it is a good question, and we do get it a lot to hear, can the device be approved for, a broad scope of drugs delivery? And I would agree that I don't think we're at that point for all the reasons that other panelists have suggested, but also you kind of bring in the complexity of, are you then approving a drug for a new indication? And are you using off-label drugs in the micro-labels, drugs that are not already approved to treat that disease, say GBM, or are you using only drugs that are approved to treat GBM? And then you bring in the point that Gautam just made of, are you increasing CNS exposure and toxicity? And so I think at this point, we really do lean towards, we have to kind of consider the drug and the device together. But, as Bennett mentioned, these are often considered on an individual basis, and these kind of things are where these pre-sub and pre-IND meetings could be very helpful if there's rationale to support, what you're trying to do more broadly, or depending on the device, the drugs, and the level of experience we have with them.
Great. Another question that came in via the chat is, so, you talked about the volume of the BBB opening being important and how you kind of have gradually been increasing that to make sure you have safety. The question is around, if you were using one device that was already in a clinical trial and you had some volume of opening, can that volume of opening kind of be translated to other devices? So I think it's sort of back to this idea of, does everything have to be done with the exact same device? Or, if you're saying, okay, there's all this great clinical data using this volume of opening, now I'm actually using a different device. Can I take what I've learned from that one device and apply it to the other device? Does anyone have any comments, Bennett?
Well, just on the top of it, as I would generally look at any number of devices, providing the performance characteristics of the device are, if not identical, but substantially similar. And we have devices that go through a path where we call it substantially equivalent, though that's for a specific class of device. But in principle, it would seem to me that as long as the performance of the device, one ultrasound device, is the same by those specific parameters that are important as another, then that might be okay. If there's some, and that's where we would certainly rely on Matt or Greg or, our expert physicists, et cetera. If there is a difference or a substantive difference, then that would, I would believe, send the situation back to the preclinical to demonstrate safety at incremental volumes that would be commensurate with similar volumes in the human. So we have, you can't look at like a mouse or a rat at 1 cc and say just 1 cc in a human. There's this extrapolation of the volume, perhaps, but but it would be based on the device performance. And that's true when we're looking at other devices that come in general, where do they have to be subject to new testing of their performance, or in a preclinical before they can be used clinically, or can we rely on the bench testing that demonstrates the similarity, or in our parlance, equivalence to an already established or cleared device. So I think that's how it would go. And I'd be interested if Matt or Greg have a comment there with regard to ultrasound per se.
Any other comments? Okay. So, we've heard some in this workshop also around liquid biopsies and the role of liquid biopsies. And so I would just like any of you to comment on where you see the use of liquid biopsies in these sorts of clinical trials.
I can comment briefly from the drug side. I think there's a lot of interesting ongoing research on liquid biopsies, and I think we would be open to having them be included in these trials, but they would generally be considered exploratory endpoints at this point because they haven't been validated to demonstrate clinical benefit from the drug side. But I certainly think there's a lot of interesting data out there in research, and so we would encourage trying to continue to explore any avenues that we could to better predict response to new drugs and new drug device combinations.
Okay. And another area of interest is around the use of focused ultrasound for inducing an immune response, or focused ultrasound BBB opening paired with immunotherapies. And so I'm wondering if you have any advice or any thoughts on how best to study the kind of systemic immune effects that are created from local treatments like a focused ultrasound. Are there any guidelines from the FDA, maybe from anywhere within the Oncology Center of Excellence or from your division, that kind of provide some information about how to really study those immune effects?
Well, I think in part it depends on what you're studying, which aspect you're studying. Are you looking at safety or efficacy of the drug and the tumor itself? Obviously, in the early stage of development, safety is a big concern of ours, and you can have heightened immune response and reaction could really be a safety concern when using the device-drug combination. And so we would be looking to make sure that was addressed in an IND, that there was appropriate safety monitoring and assessments, including stopping rules if you start to see a safety signal or have concerns based on the preclinical data that there's just going to be a safety signal. In terms of measuring, I think it probably depends on the drug itself. It depends on the target you think you might be hitting and what off targets you think you might get. So I think it's a little hard to answer broadly. I think imaging can be difficult to interpret. Obviously, we all know with immune responses that, are you seeing tumor growth, or are you just seeing immune response edema, things like that to tease out. So I think those are things to address when you're looking at response rate of the tumor. Where you have tissue, that's something you could look at before and after sometimes, if that's feasible or for the patient population. Systemically, there are also things you might want to consider depending on the drug and how much you think you might achieve. It's possible you might achieve, so PK data, and then depending on what you're looking at, circulating immune cells. And that would also be a good question for our clinical pharmacologists too early on, and what they would want to see and look for.
Great, thanks. There's also been some talk in this meeting about the value of adaptive clinical trials and more, other types of unique trial design. So I'm just curious if you think that this type of trial design would be appropriate for device drug combination trials. Gautam?
Perhaps by that, do you mean like a hybrid clinical trial design? Okay. So I think, at this standpoint, many of those trial designs may be for later stage development. And right now, I think in terms of the FUS device drug combination trials, we're still looking at earlier stage phase 2 studies, which show like an earlier estimation of activity. So that discussion about kind of adaptive trial design may be further down a conversation for further down the road.
But assuming we get to the point where we have, good safety data from several different clinical trials, and then we're really at the stage where we want to, we really want to evaluate the different combinations of all these, drugs that are out there with focused ultrasound. Is it something that you think we could get to that stage?
Jessica, are you asking, like, could you have a trial, say, where you have a common control arm, and then you could introduce different types of drugs using the same, maybe different experimental arms with different drugs in the microbubbles or with the ultrasound, and then those are all compared to maybe a common an arm that was maybe not placebo, but something that you had tested in the earlier trials, something like that.
Yeah, and I think specifically if you have focused ultrasound as the delivery mechanism, it's like it's being used for that, but then you want to incorporate and try all the different drugs. From our perspective, I think something like that would be feasible. I think it'd be a good thing to talk about early on and have everybody understand the objectives of the trial and if they can be achieved with our statisticians and everyone involved. There's a lot of variables in any trial design, but I think we'd certainly be open to something like that. Okay, great. Go ahead.
If I could just mention, this comes up in a purely IDE context for the use of the focused ultrasound at times with regard to having a comparator group. These devices allow for, in essence, non-invasive procedure, and some of the traditional procedures that the ultrasound's now being employed for are invasive. And the question always comes up, does the comparator have to be a randomized, trial, or could it be a historical comparator because of the problem with equipoise? And we continually are willing to look at that. And in fact, we understand the problems with equipoise. So again, just on the device side, we're often looking at varying ways to allow the study to proceed in a slightly more creative way because of the comparator issues.
Okay, great. And then I think we are really close to being done, but I would like to see if someone could address this last question, which is, a lot of the promising drugs to be used with focused ultrasound BBB opening are not currently labeled for GBM. So that creates a challenge in terms of what you were just talking about using the actual, drug that has some, or using the specific drug. Do you see any way to overcome this hurdle? Because this is going to be a really tough hurdle for all of the device companies that are trying to get marketing authorization for focused ultrasound therapy. So maybe just if someone has a final comment on that. Gautam.
Yeah, I think for some of the reasons we described earlier, this hurdle does have to serve some purpose, because there are certain unknowns in terms of what is the interaction between the device and the drug? What is the efficacy of that drug in glioblastoma population? And then two, what is the safety of these drugs that aren't currently indicated for glioblastoma at potentially higher drug exposure levels in the CNS? So probably some more work needs to be done, but certainly it's a promising area.
Great. I just want to thank all of the panelists. This has been a really fascinating discussion. I think, unfortunately, we didn't get to all the questions, so we'll have many for future discussions. And I'd like to encourage everyone who's participating to definitely reach out to the FDA as early as you can in your process of designing these new treatments, because they really are a fantastic partner. And I know if the foundation is going to continue to try to engage with the FDA and facilitate more opportunities like this, where you can actually engage with them and get their feedback. So thank you all again for this. Just to everyone in the audience, we're going to take a short break, technical break, and then we'll begin the next panel. So see you soon. Thank you.